Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to my respected lecturers. My name is Nofar Najra binti Muhammad Kasmira, and my matrix number is 2022800786. Today, I am very excited to share with you an interesting study titled Pollinator Sharing, Co-pollinations and Speciations by Host Shifting Among Seeds Closely Related Dioecious Fig Species, written by Su and colleagues in 2022. This research, which is supervised by Dr. Siti Karya Muhammad Hatta, offers a fresh perspective on the unique and fascinating relationship between fig trees and tiny pollinator wasps. So now, let's begin with a quick background. Did you know that fig and wasp has an incredible relationship because they depend on each other to survive? This is because fig need wasps to carry their pollen, while wasps rely on fig as a safe place to lay their eggs and raise their larvae. For a long time, scientists believed that each fig species was pollinated by only one specific wasp species, but this study has proven otherwise. It shows that sometimes multiple fig species can share the same wasp species for pollination. This is a game changer for how we understand this relationship and their role in creating biodiversity. Now, you might wondering, what were the researchers trying to figure out? Their main questions was, how do fig trees and wasps keep their unique identities if they share the same pollinator? And how does this sharing leads to the creations of new wasp species? To answer these questions, the researchers aim to understand the mechanism behind pollinator sharing and co-pollination, while also studying how this partnership impacts species diversity. Now, let's move on to the method on how research was done. The scientists focused on 6 closely related fig species that found in several countries like South China, Taiwan, and nearby areas. First, they collected fig sapple and pollinator wasps from these regions, and the fig leaves were dried to preserve them, while the wasps were stored in ethanol. After that, they extracted the DNA from the fig and the wasp, where once the sample were ready, the team extracted DNA using 2 specialized kits, the DNA plant mini kits for the fig and DNA blood and tissue kits for wasps. This DNA became the foundation of their investigation. This DNA was then studied using advanced genetic tools like MIG sequence, which are PCR-based NGS method, and the second one is ITS analysis, or known as internal transcript spacer. It refers to regions of non-coding DNA found between the ribosomal RNA, and this method helped the researchers compare genetic similarities and genetic differences between the species. SSR markers also was used for population genetics to understand how fig and wasps are divided into groups based on their DNA. Finally, they examined the mitochondrial DNA of the wasp through CO1 and CO2 sequence to trace how long ago the wasp diverged and became distinct. But it didn't stop there. The researchers applied software like Structure and Harlequin to group the fig and wasp into genetic clusters, giving them a clear picture of population dynamics. With these tools, they pieced together the complex puzzle of fig-wasp relationship were structure. Now, we are reached to the exciting part, which what did they find out from this research? First, they discovered that 5 of the fig species in South China were pollinated by the same wasp species, Blastofaga sylvestriana. This was a big surprise because it broke the old belief that each fig species has its own exclusive wasp pollinator. And they also found 2 ways in which pollination happened. The first one is sympatric copollination, where fig and wasp share the same region and the wasp pollinate multiple species in that area. The second is allopatric copollination, which happen when fig spread to new regions and establish new pollinator relationship there. Another key discovery was host shifting, where wasps switch from one species to another. For example, when Ficus erecta moved to Taiwan, the wasps adapted to pollinate this fig, leading to the formations of a new wasp species, which Blastophaga niponica. In the MIG sequence analysis, we discovered genetic differences between various fig species, which show that they are closely related to each other. For the CO1 and CO2 analysis, we found that the wasp pollinating different fig species are genetically very similar with only small differences. And lastly, the SSR marker analysis helped us identify two main groups of fig population, which were then further divided into smaller clusters. This study had some strong points. One major strength was the use of advanced genetic tools, which provided us with accurate and detailed data. These tools help us understand the relationship between species in a way that was both clear and reliable. But however, there were also some weaknesses. A key limitation was that we didn't directly observe the wasps in their natural habitats. Without seeing their behavior in the wild, we miss a chance to understand how they interact in their real environment, which could have added important context to the study. The significance of this research is clear. It shows that behavior like host shifting, when a species switch to the different hosts and pollinator sharing, when different species pollinate together, play an important role in boosting species diversity. This finding also challenged the traditional view that fig trees and their wasps pollinators always have a strict one-to-one relationship, and the study suggests that this relationship is more flexible and complex than previously thought. Moreover, the study also has limitations. Why? Because it focused on specific regions only, meaning that the result might not apply to other areas with different environmental conditions. What we found in these regions may not hold true everywhere, so the finding might not be universal. In conclusion, this research gives us a better understanding of how fig and wasp work together and adapt to each other over time. Pollinator sharing and host shifting don't just maintain their partnership, but they help them grow, creating new species and adding to the amazing diversity of life. Thank you for listening to my presentation, and I hope you found it as fascinating as I did. That's all from me. Thank you.